ASCCT, excuse me. Welcome to the ASCCT member webinar for April. We are pleased to welcome our presenter, Katie Paul. She's going to be discussing adverse outcome pathway framework to developing high-throughput assays for thyroid-disrupting chemicals. And Dr. Paul is an ORISE postdoctoral research fellow in the National Health and Environmental Effects Research Laboratory at the US EPA. So first I want to let you know the following. Just to let you know, the webinar is being recorded, as you heard. A link will be sent to members after the presentation. Also, I'm going to mute all your lines to cut down on background noise, and after the presentation, I will unmute the lines in order to allow you to ask questions. So with that, okay, Katie, I think you should be unmuted and go ahead and share your desktop.
Let me know when you can see the first slide, Christy.
Okay, I can see it and I can hear you, so go right ahead.
Great. Well, thank you first to the ASCCT and to Christy Sullivan for organizing this opportunity and for inviting me to participate. And thank you all for spending time with me today to consider using the adverse outcome pathway framework as a guide for the development of high-throughput assays for thyroid disruptor screening. Before I begin, I just want to simply acknowledge all of my coworkers who have assisted in this work, including Daniel Rotroff, Kevin Crofton, Michael Hornig, and Stephen Simmons, and in particular Joan Hedge, who has been responsible for doing a lot of the animal work with me and developing these assays, as she's an expert on thyroperoxidase, the primary subject of my talk. As many of you are aware already, an adverse outcome pathway, or AOP, is a conceptual framework to just simply illustrate existing understandings of a linkage between a molecular initiating event, i.e. the initial biochemical interaction, and an organism or population level adverse outcome. And here on this slide, I've reproduced a graphic from Gary Ankley's seminal 2010 paper on AOPs, just to demonstrate to you that AOPs are sequential, beginning on the left with the initial chemical interaction with some macromolecule, leading to cellular and tissue-based key events along a path to toxicity at the organism or population level. This is really an organizational tool that may represent relationships that are causal, mechanistic, correlative, or simply inferential. And it may highlight how well we understand chemical-mediated toxicity via a particular pathway. But in some cases, as is the case with thyroid, it might show just how complex the system might be and might highlight deficiencies in current testing strategies to target important molecular initiating and key events. The focus of my talk today will be using a network of AOPs to develop a high-throughput testing strategy to detect thyroid-disrupting chemicals. And the imperative to develop this system of screening tools for thyroid-related adverse outcomes is really evident when you consider the deleterious effects of thyroid disruption during neurodevelopment. Maternal thyroid hormones modulate fetal brain development, and so thyroid disruptor screening may identify potential neurodevelopmental toxicants. Here I'm just graphically representing a human gestation period, and the gestation week is represented along the axis at the top from 0 to 40 weeks. And what you can see is that maternal free thyroxine, or T4, is in constant supply for the necessary thyroid hormone for the fetal compartment. In the gray bars, I've illustrated fetal thyroid regulation and fetal thyroid development. So you can see that while the thyroid itself develops early in gestation, the actual development of thyroid hormone receptors and responsivity to thyroid hormone and full thyroid hormone production doesn't really reach adequate levels until parturition. Now, if I overlay the development of the fetal brain during this time, the susceptibility becomes clear. So you can see that the neural tube, brainstem, and other brain structures as well as processes to include synaptogenesis, myelinogenesis, and gliogenesis are all occurring and being modulated when maternal thyroid hormone is really providing thyroid hormone to the compartment and fetal thyroid hormone function is not yet online. Thus, women of childbearing age and developing children are the most susceptible populations to thyroid disruption, and these effects are significant and persistent. So it's well accepted in the literature that moderate subclinical decreases in maternal thyroxine on the order of a 20 to 25% change in thyroxine independent of a TSH or T3 triiodothyronine change yield irreversible cognitive and brain abnormalities in children. This correlative linkage in humans has been defined using epidemiological studies to demonstrate that moderately decreased maternal T4 or hypothyroxanemia results in decreased behavioral responses at 3 weeks of age, decreased psychomotor development at 10 to 24 months of age, delayed motor coordination and socialization at 18 months, language delays at 18 to 30 months, and small but significant IQ point losses in children at 7 to 9 years of age, demonstrating not only this correlative link between maternal thyroid hormone disruption and adverse neurodevelopmental delays, but also their permanence over time throughout childhood. So identifying chemicals capable of thyroid disruption would be essential. There's mechanistic support for this relationship as well, using rat models of maternal thyroid hormone insufficiency, and these models have been used to demonstrate that the human epidemiological data on behavior and function may in fact be due to changes in cytoarchitecture, synaptic function and myelination, and myriad gene expression changes in processes that are related to neuronal signaling and cell composition of brain structures. So how deficiencies in thyroid hormones arise is actually a very complicated set of events because thyroid hormone homeostasis is regulated at multiple nodes. And obviously, we really want to understand this complex system because of the deleterious effects. So I understand this is a busy slide, and I'll walk you through it. This is essentially a collapsed adverse outcome pathway where I've basically included several molecular initiating events that converge on common key events downstream to potentially culminate in adverse thyroid-related neurodevelopmental outcomes. So on the left-hand side of this slide, I'm showing molecular initiating events in green: thyroperoxidase inhibition or sodium iodide symporter inhibition are two events that can lead to decreased thyroid hormone synthesis in the gland. These enzymes are located in the thyroid and responsible for the synthesis of these thyroid hormones. Other molecular initiating events in other target tissues include activation of xenobiotic nuclear receptors, inhibition of the enzyme that converts thyroxine, the prohormone, to T3 peripherally, the deiodinases, serum binding protein interference, and interference with thyroid hormone receptor binding. So you can see, in contrast to endocrine systems that we typically think about, like estrogen or androgen, the thyroid hormone receptor is really one of a much larger set of molecular initiating events that essentially result in decreases in circulating T4 or T3 concentrations. Now, this decrease in systemic thyroid hormone concentrations can then lead down two different paths here shown in red. It can lead to a change in the tissue concentration of thyroid hormones or an increase in TSH, thyroid stimulating hormone in some cases. This is through a negative feedback mechanism, and this top portion where TSH is increased leading to thyroid hyperplasia and thyroid tumors is really thought to be more relevant in a rodent model. Humans are less susceptible to changes in thyroid cellularity in response to TSH. However, this bottom adverse outcome pathway, a change in tissue thyroid hormone concentration leading to altered neurodevelopmental events and neurological deficits, is relevant not only in rodent models but to humans as well. So here I illustrate on the left-hand side, that thyroid hormone system regulation with the gland at the top, circulating thyroid hormones being cleared metabolically by the liver, undergoing peripheral conversion from T4 to T3, and binding to the thyroid receptor in target tissues. And on the right-hand side, I've reproduced an image from David Reif et al.'s 2010 paper on the ToxPi visualization tool as applied to prediction of endocrine disrupting activity. Highlighted with this blue wedge are the pieces or assay information that were considered pertinent to thyroid regulation, and this included thyroid receptor binding assays and xenobiotic metabolizing enzyme assays and some other nuclear receptor reporter assays. When I highlight these particular assays on the thyroid network to the left, you can see that the ToxCast assay set included those related to thyroid hormone receptor activity and metabolic clearance of thyroid hormones, but that this left several nodes for which there are no associated screening assays, including enzymes in the thyroid gland to include thyroperoxidase and the sodium iodide symporter, which are responsible for thyroid hormone synthesis, as well as deiodinases that convert T4 to T3 to promote genomic thyroid hormone action. I've also listed here some chemicals known to disrupt these targets. TPO may be inhibited by anti-hyperthyroid drugs, including methimazole and propylthiouracil. More environmentally relevant to us would be compounds like benzophenone 2, which is a UV blocker found in sunscreens, and dietary isoflavonoids like genistein and daidzein. The sodium iodide symporter, or NIS, may be inhibited by anions or some small organic cardioglycoside drug candidates. Thus, it's apparent that we need more assays to simply cover all of these potential molecular initiating events for thyroid disruption. And if we had these assays, we might be able to build a more predictive ToxPi for endocrine disrupting activity to better include thyroid disrupting activity. So I've chosen to start with my assay development endeavors with the thyroperoxidase enzyme target for which there was not a medium or high throughput assay. However, there was a legacy assay, and I'll tell you a little bit about that before I tell you what I've done to create a high throughput version. This assay that I refer to as the legacy assay is the guaiacol oxidation assay. It uses guaiacol as a substitute for diiodotyrosines in the coupling reaction that's catalyzed by thyroperoxidase to generate thyroid hormones in the gland. So guaiacol is coupled by thyroperoxidase to a diguaiacol product in the presence of hydrogen peroxide, and then this diguaiacol product may be measured at 450 nanometers on the visual spectrum. The guaiacol assay, however, is limited in its screening utility. It's time-consuming to run, as only 3 to 8 wells of a 96-well plate may be performed and read at once due to the rapid reaction time, which must be monitored over 60 seconds. This method also uses quite a bit of rat thyroid tissue as the source of thyroperoxidase. So you can test about 12 reactions, 12 rats would be needed for 96 reactions. So we'd like to extend the amount of testing we can do per rat thyroid and reduce animal use. And finally, we'd really like an assay that wasn't kinetic. So we'd like a stable signal. Part of the problem with the guaiacol assay is that the diguaiacol product is not stable. It can revert to guaiacol or be further metabolized, and that's why only a few wells of a 96-well plate can be performed at once. So the goal was to replace this chemistry with something more suitable to screening. And so what I've done is use Amplex Ultra Red in place of guaiacol. Amplex Ultra Red is a peroxidase substrate that's available commercially from Life Technologies, and essentially in the presence of hydrogen peroxide, thyroperoxidase will convert Amplex Ultra Red to resirufin. This would be an endpoint assay where resirufin has been shown to be stable in my hands from 30 minutes to at least 2 hours post-initiation of the reaction. And this can be read on a fluorescence scale and is quite sensitive. Interestingly, Amplex Ultra Red was marketed for use with horseradish peroxidase to detect hydrogen peroxide in cell-based systems. So you can see we're using this sort of off-label for our purposes for screening. And this really is only possible because of conservation of catalytic domains of mammalian peroxidases. So what I've done is try to develop and optimize an assay with Amplex Ultra Red in place of guaiacol, and to use methimazole, a positive control, to demonstrate the assay performance. I've varied protein concentration, Amplex Ultra Red concentration, the assay buffer and its contents, the plate type to include black-walled, white-walled, clear bottom, clear plate. I've characterized DMSO sensitivity, and I've also looked at reading this assay over time. So I'll just review for you the results of all of that, which it's really funny to encapsulate on one slide so much of one's life, but it was a really fun process that really started with characterizing the original legacy assay shown here on the left-hand side of the slide. I represent the guaiacol assay as GUA, G-U-A, and then I developed the new Amplex Ultra Red assay in a 96-well plate first, and then scaled this to a 384-well format. So I realize there's a lot of data here, but I'd like to walk you through some of the typical assay metrics that I would use to grade an assay when I'm trying to develop and optimize it. So you can see I've listed plate format in accordance with the graphs that they correspond to. And then dynamic range is the first metric that I've included. You can see that even in a 384-well plate format, the Amplex Ultra Red assay with a positive control has a dynamic range of about 11.5-fold control, which is quite good, and we have some confidence we'd be able to resolve a signal from background. Z prime here is shown. Z prime scores for both the Amplex Ultra Red assay in 96 and 384-well formats are excellent. For those who are less familiar, Z prime is derived to understand how many standard deviations separate the positive and negative responses in the assay. So a Z prime of 1 is the theoretical ideal and cannot be exceeded. You can see in a 384-well plate, our Z prime score is 0.9. The coefficient of variation on the legacy guaiacol assay was quite high, as you can see. This really owed to large variation around the vehicle control data and the top signal data, and the coefficient of variation for both the 96-well and 384-well formats of our new assay are within acceptable metrics. This is a really interesting line in this table I'll spend a minute talking about. So MMI is my abbreviation for methimazole, and you can look at the 50% inhibitory concentration, or IC50, and for the guaiacol assay, it's about 2.2 micromolar. For the 96-well version of Amplex Ultra Red assay, it's 0.09 micromolar. And for the 384-well format, it's an even greater potency, i.e. a lower concentration of 0.025 micromolar. And I think this really owes to optimization steps. So I've drastically reduced the amount of protein present in each of these formats as you move from left to right. So in the guaiacol assay, it required 50 micrograms of rat thyroid microsomal protein per reaction. The 96-well plate version of the Amplex Ultra Red assay used 12.5 micrograms, so we've reduced the amount of protein by a quarter as we move across. From 12.5 micrograms of protein, I reduced this to 5 micrograms of protein per reaction in the 384-well format. So we have a 90% reduction in animal tissue use from the guaiacol 96-well format assay to the 384-well format assay of the Amplex Ultra Red TPO assay. And this is reflected here in the final bar in this table, where you can see previously we could get about 8 endpoints per rat. Now we can obtain about 83 data points per rat. This assay method is amenable to screening due to its optimal logistics. It's performed in a 384-well format. It's addition only, meaning that reagents are added and not aspirated or taken away. I use automated liquid handling, and the reaction can be read as an endpoint and is stable from 30 minutes to 2 hours post-initiation of the reaction. So I dispense the rat microsome dilution pool containing buffer, and then I use liquid handling to dispense the Amplex Ultra Red. I dose these biochemical reactions using a high-density replicating tool just to automate the chemical administration. And then I dispense hydrogen peroxide to initiate the reaction using liquid handling. The plate is incubated in the plate reader for 30 minutes at 37 degrees. It also works at room temperature. And then the plate can be read at 30 minutes, or it can be read 2 hours later. So if you had a stack of 384 well plates to read on your reader, you could actually get through them and not worry about the signal changing. So before I advance to the next slide, I just want to point out that this would basically enable testing of more chemicals than had ever been evaluated for thyroperoxidase inhibition. But before we test a set like that, we would really like to know how the assay would perform with something other than a positive control. So we needed to demonstrate how the assay would perform with a diverse set of training chemicals. I selected this 21 chemical training set based on data from the literature and previously tested chemicals in our laboratory. In red, I've highlighted the 12 anticipated positive chemicals. I've also included 9 anticipated negatives. I won't describe each of these in detail for now, but briefly, the positive chemicals included anti-hyperthyroidism drugs, methimazole and propylthiouracil. It included the rubber vulcanization agent that's a byproduct of tire production, this mercaptobenzothiazole. It included estrogenic phenolic compounds, including propoxyphenol and nonylphenol. Benzophenone 2, I've already mentioned, is a UV blocker found in sunscreens. Resorcinol is a coal pollutant. Ethylene thiourea is a common metabolite of the bis dithiocarbamate pesticides. Amitrole is a herbicide. Daidzein and genistein, many of you will be familiar with as dietary isoflavonoids. And then sulfamethazine is a sulfa drug, a sulfa antibiotic. So I want to give you a snapshot of the results of this 21 chemical training set. First, all of these plots of chemical data are percent of DMSO vehicle control on the y-axis, normalized to 100%, versus methimazole or chemical concentration on the x-axis. The 3 symbols present at each concentration, the diamonds, circles, and squares, represent distinct biological replicates, and each biological replicate was performed on a different day with a different lot of microsomes. And Joan Hedge and I prepared bulk lots of rat thyroid microsomes with 60 rats per lot, so there's a lot of biological variation encompassed in each of these points. This slide I'm showing the TPO inhibitors, or positive chemicals. All chemicals were evaluated using the same concentration curve, which ranged from 50 picomolar to 253 micromolar. 11 chemicals dose-dependently inhibited TPO, I'm showing here in red, and 3 additional chemicals inhibited TPO at the highest concentration tested. The chemicals that tested positive included all 12 anticipated positives from the literature, plus 2 additional chemicals that were previously reported as negatives, triclosan and iopanoic acid. They're some of the more equivocal positives here, as they had a very weak response in the assay. Both of these chemicals are actually known to decrease thyroid hormone concentrations in rats in vivo, but are thought to act via other modes of action. So the mode of action for triclosan for thyroid disruption was actually the subject of my dissertation, and we showed that it upregulated markers of hepatic catabolism of thyroid hormones. And iopanoic acid inhibits peripheral deiodination of T4 to T3. So we can't really say that either of these compounds are necessarily false positives, as they decrease thyroid hormones in vivo, and study of their impact on TPO has been limited to one other report with the legacy guaiacol assay. However, it should be noted that these compounds only inhibited TPO activity at 253 micromolar, and triclosan would certainly be cytotoxic in a cell-based model at that concentration. So while they may not be false positives, they may be irrelevant. Importantly, I do want to highlight a few distinctions in this training set. First, benzophenone 2, as we've discussed, was a positive, but interestingly, the structurally analogous BP3, another UV blocker, was negative. So now I'm showing you the negative chemicals with no effect on TPO activity. BP3 was one of them, so we're able to distinguish between structurally similar compounds with different activities. Phthalate compounds that I tested, including diethylhexyl and diethylphthalate, were negative. This methyl-methylbenzoate compound was negative and is known to be negative in the amphibian thyroid explant assay. So it's a cross-assay negative compound. And here, sodium perchlorate, Na-per, is known to inhibit the sodium iodide symporter and not thought to act on TPO. So we can distinguish between chemicals that act at other targets. Previously, DPM was reported to inhibit TPO, and actually sodium perchlorate has also been reported to inhibit TPO, but both of these chemicals only act at concentrations in the millimolar range, which exceeds the range that we tested and likely exceeds the range of what you could potentially achieve in a thyroid gland. So just to review the success that I think we've demonstrated with the 21 chemical training set, the 384-well plate Amplex Ultra-Red TPO screening assay used rat thyroid microsomes as the source of TPO, had a stable fluorescent signal, were highly reproducible, evidenced by Z prime score, and had adequate dynamic range to identify TPO inhibitors, and represented a 90% reduction in animal tissue required per well tested. The IC50 values for known TPO inhibitors demonstrate greater potency, i.e. they act at lower concentrations in the Amplex Ultra-Red TPO assay. And this would suggest an increase in sensitivity from the glycol oxidation assay and a potential increased ability to detect weak inhibitors, but more would have to be done here for me to feel certain that that's the case. And finally, just to kind of point out how successful this is, the assay demonstrated a 100% true positive rate and a 0% false negative rate, just with the side note that there was one previous report of a positive result with 2 of the chemicals tested far above the concentration range that we tested. And just to hit home on the 3 R's, this work certainly supports efforts to refine and reduce the use of animals in toxicity testing. And later on this talk, I'll show you some pilot data on the possibility of eliminating animal use for TPO screening. So with this reduction in animal use, it really increases the feasibility of screening at the thyroperoxidase target. So not only do we have an automated reproducible assay, but we also have the ability to generate enough thyroid microsomes in a short amount of time to run a screen of a test set. So here I'm just showing you what I've already told you, that we've basically moved from needing 50 micrograms of protein per reaction to 5. So this means that we could screen a larger chemical test set. And here I'm just showing you a few different scenarios that I've come up with a mock idea of how many rats would be required, for instance, to screen the ToxCast E1K, or about 1,000 chemicals that are of interest for endocrine disrupting activity. If we just did a single yes or no format, since this is an inhibition assay, we could use 1 high concentration for each chemical, do 2 technical duplicates and 3 biological replicates on all 1,000 chemicals. And this would mean about 72 rats, which for Joan and I is about a day's work. We can create the thyroid microsomes and then bank them in the freezer until use. So is this method actually ready for use in thyroid disruptor screening? This is a very busy slide that I won't actually read all of it to you. This is a table that I've adapted from a paper that Kevin Crofton et al. Published in Alltex in 2010, where they were trying to describe guidance criteria that would be useful in the development of developmental neurotoxicity high-throughput screening tests. So you can see I've highlighted just 3 of the guidance criteria because I want to speak to these a little more regarding the Amplex ultra-red thyroperoxidase inhibition assay. So first, key event, how well are we modeling a key aspect of the thyroid system? Well, what we'd really like to know is that in vitro TPO activity is relevant to in vivo thyroid hormone disruption. And I have some pilot data on this that I'd like to show you. Second, response characterization. This is the level of change determined to be in effect. Currently, with this high throughput model, I've arbitrarily set the threshold for effect to a 25% change. However, what I'd really like to do is understand how much TPO has to be inhibited in order to mediate a 20% decrease in thyroxine in vivo, since that is the jumping off point for considering maternal thyroxine decreases adverse. So we'd really like to anchor this to physiology, and I'll show you a little bit of data related to that. And finally, transferability. So we'd really like a high-throughput screening assay to be openly available for any laboratory to run. This assay is transferable, and Amplex Ultra-Red is commercially available, but thyroid tissue is slightly difficult to obtain if you're not used to performing that kind of necropsy, and it requires specialized expertise. And so I've been working on a way to replace rat thyroid tissue use in this assay. But first, what does in vitro TPO inhibition mean for an organism, i.e. how well do we model a key event? So what I'd like to do is take rats that we expose in vivo to methimazole, measure their circulating thyroid hormone concentrations in vivo, and then remove their thyroid glands and use them to make microsomes to then test in the thyroperoxidase inhibition assay. This would give us what I'm calling ex vivo thyroidal TPO inhibition. We could then potentially relate this to what the concentration of methimazole was in the thyroid gland in juxtaposition to what the oral dose was for methimazole. We could develop a relationship between this ex vivo thyroidal TPO inhibition and in vitro TPO inhibition, so when just microsomes are exposed in a dish. Ultimately, the relationship we're really interested in is human methimazole-induced thyroperoxidase inhibition in order to build a model understanding of how rat in vitro methimazole TPO inhibition relates to human recombinant TPO inhibition. So I'd really like to start developing an in vitro to in vivo model for TPO. We've started to do this with the rat, and I'll show you just some pilot data. Methimazole has been given to rats many a time in the literature. This is our particular take on the assay because we're measuring endpoints that aren't always reported. I'm showing you data from a 4-day exposure in rats. So the graph on the left-hand side is the thyroxine dose response. So on the y-axis, it's percent of control T4. And on the x-axis, I'm showing increasing methimazole concentration or oral dose given per day. And you can see with increasing oral dose of methimazole, thyroxine in the serum is decreased. On the right, I'm showing thyroid gland weight versus methimazole dose. And on the y-axis is just percent of control, thyroid gland weight. And you can see with increasing methimazole dose, there's increased gland mass. This is really due to hyperplasia that develops in response to the decreased T4 and T3 from methimazole exposure. Now I'm showing you on the bottom Amplex ultra-red thyroperoxidase inhibition activity in the thyroids from these animals. So this is the ex vivo thyroperoxidase inhibition data. What you can see on the left-hand side on this y-axis is percent of vehicle control, Amplex ultra-red TPO activity, and increasing methimazole oral dose exposure since we did not have a measurement of the thyroid gland concentration. But just using oral dose, you can see that with increasing methimazole exposure, Amplex ultra-red TPO activity is decreased. So we have a consistent response where we saw thyroid hormone disruption in vivo, and it corresponds to amplex ultra-red thyroperoxidase inhibition in vitro. So if I just draw the IC50 for amplex ultra-red TPO activity on the graph, it is predicted to come at about 228 milligrams per kilogram per day of methimazole. Now, if I look at that dose on the T4 and thyroid gland weight curves, there would be about a 40% decrease in T4 at that dose and a 60% increase in thyroid mass. So we see something that looks like a correlation here, but we'd like to repeat this study in a full study mode because we have a little non-monotonicity of the data that we'd like to verify. So you can see here in the T4 data, there's this appearance of an increase. We're not really sure that this is biologically relevant. And if the data were monotonic, it might be easier to develop a correlation. So on this next graph, I show you my effort to try to correlate amplex ultra-red TPO activity with T4 concentration on the right, on the left, and amplex ultra-red TPO activity with thyroid gland weight on the right. So you can see amplex ultra-red TPO activity and T4 do not form a statistically significant correlation. However, the thyroid gland weight, which has a normal dose response curve, does correlate significantly with amplex ultra-red activity. But obviously, we would prefer a statistical correlation with thyroxine because a decrease in thyroxine is typically what might be measured in an epidemiological study or even in a rodent study. So that's really the physiological benchmark we'd like to use for a correlation. So we'll repeat this study and try to develop this physiological anchor for the high-throughput screening assay in order to understand when we inhibit thyroperoxidase in an in vitro screening assay by 50%, what kind of T4 decrease might that correspond to in the animal? So in considering rat thyroperoxidase inhibition, obviously a question that a lot of people have for me is how appropriate is this for predicting human health hazard and the human key event? And the answer is rat TPO is a good surrogate model. So here I'm showing you just a visual representation of an amino acid homology experiment that I performed. There are 6 putative domains for the human thyroperoxidase enzyme. And if you just do an amino acid sequence alignment between species, what you find is that the putative catalytic domain has high sequence identity across species. So in each row, I'm showing a pairwise comparison. So this is human to porcine. Porcine TPO is used in a lot of legacy glycol assays. Human to rat, and then rat to porcine. And you can see that they all have around 80% amino acid identity. So this suggests that rat TPO is a useful surrogate for human TPO, and this is a screen that would be worth doing. However, we would like to replace rat thyroid tissue. It not only makes a more relevant model when we're considering human risk in the sense that we'd like to use human cells and human tissues, but it also would make this assay more transferable and eliminate animal use altogether. So what I've done is I've transduced human embryonic kidney cells, HEK293Ts, with a lentivirus that I've designed to contain a human thyroperoxidase expression gene, a hygromycin resistance gene for antibiotic selection, and a green fluorescent protein marker for just confirming cellular transduction. And then from this cell line, I've generated cell-based microsomes similar to our use of rat thyroid tissue and used them in the amplex ultra-red thyroperoxidase assay to demonstrate responsivity to methimazole, our positive control. And the preliminary data are quite promising. I'm showing you some of the pilot data on the left-hand side now. On the y-axis is percent of vehicle control activity for amplex ultra-red TPO. And on the x-axis is an increasing concentration of methimazole. This was done in a 96 well plate format for assay development purposes. You can see that the black bars, which correspond to the cells that would be expressing human TPO, there's a response to increasing methimazole concentration. So there appears to be TPO activity that is inhibited by methimazole exposure. The white bars are the HEK293T wild-type cells, which I'm just showing their background activity. So you can see we have a dynamic range here. Depending on the run, it varies from 3 to 8-fold vehicle control. So it appears likely from 96-well plate pilot testing that human recombinant TPO could be used in place of rat thyroid tissue for screening. And I think that is what we will do next. So our anticipated future work with this assay, we'd really like to test a larger set of chemicals, whether it's phase 1 or phase 2 of ToxCast, the E1K, or all of the above. This would be extremely novel because no one has tested that much of chemical space for thyroperoxidase inhibitor activity. If we did this in a yes or no format with just one high concentration of each chemical, we could do follow-up dose response work on the test set positives and equivocals. We could include other assays in parallel as well. We could try to isolate nonspecific protein inhibition by running a luciferase inhibition assay just to see if a chemical, something like a detergent, might be inhibiting protein nonspecifically and not have a true affinity for thyroperoxidase. We'd also like to contextualize the responses in these assays. So cell viability data will be critical. Since this is a biochemical assay, we can often obtain concentrations much higher than what would be tolerated by a living cell. So we might want to run a thyroid cell line or even just a HEK293T cell viability assay to contextualize this data and look for what's relevant. I'd like to finish optimization and a training set testing with recombinant human TPO as a source. But ultimately, what I want to talk to you about for the rest of our time here as it's coming to a close is how does this relate to a concerted effort to develop a model of thyroid disruption and screening for thyroid-disrupting chemicals. So back to my busy adverse outcome pathway. I've placed some green check marks by molecular initiating events for which we have some screening assay coverage. In a red box is the sodium iodide symporter, which is the target for my next assay development adventure. But what you can see is that this leaves molecular initiating events with no assay. So what I want to do is juxtapose this AOP on the next slide with a testing strategy, if you will. So this is my simplified testing schematic that I would propose that would involve an initial battery of screens that increasingly converge on tests of increasing biological complexity that reflect the adverse outcome pathway. So on the left, in place of molecular initiating events, I have boxes now representing available high throughput or medium throughput screening assays. The green boxes represent molecular initiating event related assays that are currently available. So we've just developed this Amplex Ultra Red TPO screening assay. It's now available, but there aren't good assays available for sodium iodide symporter screening, cellular influx of thyroid hormones, or deiodinase inhibition. And just as a reminder on the bottom of this slide here, I'm reminding us of the deal that we make in covering chemical space. So when we have a high throughput screen, we can cover a lot of chemical space, but these assays typically are of limited biological complexity. But as we move to the right and encounter confirmatory tests that encompass related key events, you can see these assays will have increasing biological complexity. So here I suggest that thyroperoxidase screening and sodium iodide symporter screening assays that converge on the same set of confirmatory tests. This could be an amphibian thyroid explant culture, which would require TPO and NIS functioning in order to visualize production of T4 in the explant. It could also include a mammalian cell culture model, which I have here in this purple box. And TBI is my shorthand for to be invented, which is something I'm interested in working on. Here I show you that nuclear receptor reporter or binding assays, cellular influx of thyroid hormones, and deiodinase inhibition could converge on similar assay platforms. They could use primary hepatocytes or microsome-based assays. However, primary hepatocytes and microsomes too, these assays would be performed at different rates because these might be transcriptional, they might be functional, so the throughput on these assays would be varied. And then here I'm just representing that there are some confirmatory tests for serum protein binding and its high throughput screen, and thyroid hormone receptor binding. We could do radioligand binding assays, for example. So we would have a battery of confirmatory tests following high throughput or medium throughput screening that would then converge on really the same targeted testing animal models to measure the apical adverse outcomes in the individual. So all of these could converge on a 4-day rat model, as I showed you with our methimazole work. This is a model that we've employed and others have employed a lot as well. Or the 7-day amphibian assay, which has been developed by Michael Horning and Joe T.G. And Sig Digetz at EPA in Duluth, Minnesota. And then from there, we could move on to susceptible populations, so the 21-day amphibian metamorphosis model or a developmental rat model where the developing rat would be exposed to a thyroid disruptor. So that is the goal, and keeping that in mind, we would really need more assay development is what that makes clear. And so I've already intimated to you that the sodium iodide symporter is my next target of interest for developing another screening assay. I've identified candidate chemistries in a couple of models to try to demonstrate sort of an initial jumping off point, and this might incorporate the use of cells that express the sodium iodide symporter endogenously and then moving to an engineered cell line. So you can see this is an iterative process where we might start with a legacy assay or a medium throughput assay and then try to tinker with it to develop an assay that is more appropriate for high throughput screening. So just to give you a small preview, I'm really interested in this candidate chemistry, the Sandel-Kolthoff reaction, whereby yellow cerium is reduced in the presence of iodide to a clear product. So essentially, if the sodium iodide symporter is functional and taking in iodide into a cell, then that iodide will reduce the yellow cerium to colorless cerium. So I could develop potentially a functional assay with rat follicular tumor cells or FRTL5s that express the sodium iodide symporter, first in a 96 and a 384 well plate format, just to try to get the chemistry working, get the assay working. And then we could extend this assay to a human recombinantness model using lentiviral transduction of a cell line like human embryonic kidney cells. And then finally, I'd really like to experiment with replacing the chemistry once the model is functional with another substrate that forms a stable product when exposed to iodide. What I kind of glossed over is that this reaction occurs also in the presence of arsenious acid, so arsenic. So it's not necessarily appropriate for high throughput screening, might be appropriate for medium throughput until we can develop a cell model and then replace the chemistry. So my final conclusions are that more assay development for molecular initiating events related to thyroid AOPs is needed for an integrated screening approach. If we'd like to develop a computational model of thyroid disruption or a better predictive systems biology tool like ToxPy, we would really need assays that cover each molecular initiating event because these events may occur independently of each other. They don't necessarily have any relationship with one another. We might also need duplicative assays at these biological nodes in order to develop a reliable model. And I think this assay development effort will be extremely critical as we move forward with EDSP21 efforts because these efforts have focused predominantly on estrogen and androgen signaling. And for thyroid hormone disruption, we really are at a basic science state where we're trying to evaluate what can be developed into a high throughput screening test in order to build predictive tools. So with that, I'd like to acknowledge everyone that I've worked with at the EPA on this project to date, including Joan, Michael Hornig, Steve Simmons, my mentor, William Mundy and Danielle Ackerman, the technician in my lab. I also work very closely with the Center for Computational Toxicology. I've worked with Dane Filer and Daniel Rotroff to refine my R scripts to analyze this data. And finally, funding. I received funding from EPA and the Oak Ridge Institute for Science and Education Fellowship. And here I'm showing you a little movie of our high-density replicating tool. This is how we automate the dosing of all of the different assays that we run in our lab, which are many. We have quite a portfolio of assays that we run, not just endocrine, all different endpoints. And this really makes it possible, as administration of the chemical would be the most time-consuming part of doing this if you did not have automation. So with that, I would be happy to take questions.
Okay, thank you so much, Katie, for that presentation. I'm going to unmute everybody's line. And if you have a question, please just speak up. Well, I will just point out, I thought it was really interesting. You had a slide of your proposed sort of an ideal testing strategy or potential testing strategy. And the amphibian metamorphosis assay, which is your very last animal assay, like the furthest tier, is what is considered a tier 1 screening assay for the endocrine disruptor program.
Right, and so my idea is to kind of back engineer a high throughput screening system before going into that model, because that model really can't be used to test thousands of chemicals, and we need data on thousands of chemicals.
Yeah.
Yeah, it is kind of an inverse view of what we're used to seeing, where for EDSP, the 21-day amphibian is a tier 1 screen. I'd like to back engineer screens before that.
No, I think that makes a lot of sense, and is really the way we should be doing things. I just thought it was really ironic. Anyone else have a question?
Yeah, I do, Christy. It's Pat Bishop from PETA. Hi, Katie. It was a great presentation. I really enjoyed it.
Thanks, Pat.
You did mention at the end, EDSP21, and, we've all been sitting on our edge of our seats here wondering, how quickly that's going to be able to move along. And we did see some presentation on the estrogen computational model early in, I think it was January. EPA folks had a SAT meeting and talked about it. And there was still some bumps in the road with that, but they seemed like, they were moving along on it. And I guess androgen, it sounds like it's not too far behind. But, from your presentation on thyroid, it seems, obviously a much more complex system, and you still need a bunch of assays. And I was just kind of wondering, first of all, are you the only one really working on this to kind of move this into EDSP21, or whether you are or not? What do you think might be a point in time where we might be able to see this being used to start to replace some of the tier 1 assays?
Well, that's a great question. I'm not sure I can speak to all of it. So within the National Human Environmental Effects Research Laboratory, this is the only research effort to develop high throughput screens. So it is a limited effort with limited resources, but not due to its importance. I think it's extremely important, and it's garnering more attention now, I think, like you said, as estrogen models are becoming more clear, although they are more complex than we initially thought. It's a little bit more complicated than just the estrogen receptor that we typically think of, but I think more and more people are starting to get a handle on a predictive computational model based on high throughput screening data for estrogens. Thyroid is much more behind. We're missing methods. I'm not sure when this would or could replace the tier 1 screening assays. That's really, I guess, a regulatory question. But my sense is that someone needs to step forward and present models like this, where we propose what to do with all of the chemicals, and then how to advance chemicals that are positive or equivocal into confirmatory tests that have increasing biological complexity. But I think that until there's better recognition of these assays and better understanding of these assays in a high throughput scenario, it won't be possible. So my hope is to run a larger test set through the Amplex Ultra-Red TPO screening assay sometime before the end of 2013. It involves coordination of a bunch of different groups, within EPA to get the chemicals and test them. But I think this would be important because then we can start to show that we have a plan for what to do. So I actually have a separate, more detailed plan of what happens when we screen with the Amplex Ultra-Red TPO assay. What are the follow-up assays that need to be done? How do we deal with negatives, positives, equivocals? And so these decision trees are really important, and I think once we evaluate at least one or two modes of action within this complicated adverse outcome pathway using high throughput screens, it will become more plausible that this could replace the Tier 1.
So it sounds like it's still a few years down the road anyway at this point.
Well, if I had more money, maybe it'd be closer. I don't know who can help me with that, but yeah, I'm a postdoctoral fellow, so this is really my research interest and area, and I'm trying my best to develop as many assays as I can while I'm here.
Mm-hmm. So you're kind of on loan to EPA, or what is your relationship with EPA?
I have a contract relationship, so I receive a fellowship from the EPA that's administered by Oak Ridge. So it's just a funding vehicle for postdoctoral fellows who aren't federal, because those positions are few and far between now.
Yeah.
Most fellows are coming in on some sort of contract vehicle, and that's all that is. But it does mean that you're very temporary.
Anyone have any last-minute questions? I have a really quick science question for you, actually. So when you were talking about linking the dose in sort of the quote-unquote dose of the chemical to the in vivo thyroid gland concentrations, as you talk about doing that for either in rats or in humans even, is there a lot of inter-individual variation in that metric, or do we have a handle on that?
So I am not sure. We typically, people who take methimazole or animals that are given methimazole in a veterinary setting don't have their thyroid gland concentration of methimazole measured. So typically a clinician will just titrate the methimazole until their thyroid stimulating hormone is in the correct reference range. So it's really not executed in a clinical setting in a way that would allow us to get the measurement we really are interested in, which is the thyroid gland concentration. Because the oral dose and the thyroid gland concentration, there may be inter-individual differences in metabolism and distribution that would cause differences in tissue concentration, but that data is really not available. Most people don't donate their thyroid in the clinical setting, unfortunately. So this is something that I would like to do in the rat model, which may or may not reflect the human variability very well, but it would give us a better idea than just oral dose. So that's ultimately the measurement that needs to be made in these sort of physiological anchoring experiments.
Great. Well, I think we're out of time. So thanks again for participating, and thanks again, Katie, for the presentation. It was really comprehensive and interesting.
Thank you.
Okay, I hope everyone can join us next time. Bye-bye.
Thank you. Bye-bye.